Hello.
Hello, good afternoon. It's afternoon here. Could you hear my voice?
Yes.
That's good.
So how is it going? Is everything all right?
Yeah, we have a slight mishap, but it's already finished. We could not use the license for several hours. Apparently, the problem is the Wi-Fi. Our Wi-Fi, our university Wi-Fi blocked the connection
I see.
for Tobii specifically, which I don't understand why. But there are several things that we would like some help with.
Okay. Did you manage to install the software?
Yes, I managed to install the software and get it running.
Mm-hmm.
We we tried to use it several times and it works
Okay.
fine. But we would like to understand how to get the data that we need.
Yes.
We to, we would we would like to do a research about
Mm-hmm.
educational research. So, excuse me, what's my name? I'm Agung. I'm nice to meet you, Mr. Kiro. We are from UNESA, and
Okay.
unfortunately, the our leader, Professor Nadi, is unable to join at this moment. He will
Right.
join later uh, because he has some meeting with rector and other professor. Therefore, we are the team member, and we would like to ask several things about the topic.
All right.
Uh, okay, I will start. So we plan to use the topic to measure the awareness, the concentration of students when they
Mm-hmm.
are uh, doing some homework or tests. But
Okay.
and for that we need to understand. We need to see how long in uh, the participants stare at the question. For example, the participant stare at the question 5 for several seconds, and then he moved
Okay.
to work on his uh, trying to solve the problem, and then you move to question 6. And how do we get that data?
I see. Do you have some kind of an application or like I don't know what is it, in what form are your tests? So how do you want to design your experiment? Is it just like, a picture maybe with the question, or is it like an HTML file or some other application? So
Yeah,
how does it
we have
look?
to do it in page like PDF. So in
PDF.
PDF we have the question, and then they do the work in paper mode, and then they just go to the question, and then they work on the answer question and answer, and we would like to measure that. We could get the data for the coordinates of what they're seeing, but we could not get the data for the duration. in this second, where's the eye is looking at, etc.
I see.
I
Okay, so
wonder.
I understand. So you mentioned that you already tried the software. So which software did you try? Was it Tobii Pro Lab or iTrack Manager?
We installed the Pro Eye Manager and the Tobii Pro Lab, but
Okay.
uh, Mas Dimas, can you help me? Which one? Do you mind if I share my screen?
Yeah, of course.
Okay, not this.
Good.
Uh,
Attention, attention.
yes. Okay. Untuk data, output datanya.
Oh, output datanya di komputerku, Pak.
Mhm.
Okay. This is our picture
Mhm.
for Jason and
Yeah, I see. Okay, so you just edit pictures of your study.
sorry.
You just edit a
Oh yeah.
picture with some task. So, but what about the PDF that you mentioned?
No, we haven't used the PDF yet.
We haven't used the PDF yet.
Hmm.
We still using picture
Hmm.
for testing and try the application.
Okay, I see.
Ah,
So what I would suggest, like we anyways have a slightly different focus today, but what I would suggest, if you have a moment, if you can share your PDF with me and maybe the project that you created, I could just probably try to guide you a little bit on my end, but it will be easier if I see what you are doing. So if you can just send me your PDF that you wanted to try, because the thing is that we don't really have a ready solution to run studies with PDF. You will have to do a screen recording. Either screen recording or you need to break it down to certain pages, like page by pages. So um, but it will be easy if you could send it to me. If it's not in secret, of course, just for the training purpose.
So ini sulit dicapai.
So, do you think you can share it with me, or is it not possible?
Yeah, it's still in process.
Okay, yeah, you but like take your time. We can already start with the training. I don't really need it now. I still have a couple of questions to you in the meantime. Have you already worked with eye tracking before, in your view?
Yeah.
Oh yeah, we could start with the training, please.
Yes, yeah. But my question was like, have you ever worked with eye tracking before?
Mm, Personally, I haven't at all. We've
Well,
been doing some practice in VR, AR, but eye tracking is really new for us.
okay, so for all of you, and you are doing educational research in general.
Yes,
What is it? Okay.
UNESA is the focus of UNESA as a university is in educational.
I see. Okay. So the plan for today is I'm gonna, well, let's put it like this. I have a presentation for you, and I will start like very low level then. So we'll cover some attentional aspects of eye tracking, then some technical aspects, then we will talk about your eye tracker and also like what are the good practices of working on it with it. And this will take probably maybe an hour and a half. So basically the biggest part of today. After this, we will try the eye tracker and talk about your experiment specifically. Maybe we manage to design it. So I will help you, like, designing it, and we decide on maybe some data collection task for you. So then we have our second session. By the second session, you should make some recordings, right? And we can focus on the analysis part. And this will be mainly for the next part. But then you will make some recordings, you will send them back to me, you will have time to kind of look into stuff, maybe prepare some questions. And the main focus will be, just the analysis and other things that you want to focus on. So today it will be more like, introductory part. Do you think it sounds good, or do you have any like specific requests?
Yeah, that sounds excellent, sir.
Okay, let's do so. We have like roughly 2 hours, and let's focus. Maybe we will make a short like coffee break in the middle. So just because, yeah, just a quick one. All right, let me share my screen, and then we can start. Like, if you manage to send the files during the training, just let me know. I will check them, but we don't have to wait. Cool. Let me just share my. Sorry, I'm a little bit sick today, so I had to work from home today. But I'm well set, so I have everything for the training. Cool. Mm. Right. So my name is Kirill Novikov. I'm a research scientist at Tobii. So basically, I work with researchers like on all levels, which includes trainings, consultations, webinars, educational materials, and also I participate in let's say commercial studies as well. So during the training, like, please, if you have questions, just let me know, just speak up. So don't pile your questions up, and then I will decide either it's worth like answering right away. If it's not a part of the slide, so I will just tell you that, well, we'll cover this part, but please just ask if you have any questions or comments. Oops, sorry. So why do we even do eye tracking? Well, obviously different research groups will answer differently this question, but the main thing is that understanding where we look and how it influences our behavior and cognitive processes, like which includes obviously like memory, attention, decision making, and all of this, which also applies to your type of study. So there is a lot of cognition going on in learning. So by understanding where we look and how it will influence our cognition and behavior is a fundamental question, and quite a lot of disciplines, which includes psychology, neuroscience, and also like quite a lot of connected fields. Let's put it like this. The main, or like let's say most eye tracking studies are based on so-called eye-mind hypothesis, which states that eye movements indicate cognitive processes, and if we track these eye movements, we can draw conclusion on brain activity and those cognitive processes. So basically, there is no appreciable lag between what's being fixated and what's being processed. So to make it simple, what we look at is in most cases what we think about. And we kind of can understand what's going on in the process of thinking. But we still need to start with human vision very basically, because we're working with eyes. This or that way we need to understand how we process information. And we need to start with the eye, because that's where, like, where everything starts. So this frontal surface of the eye is called cornea. It's just like, to separate the interior of the eye from what's on the outside. But the main why, the eye contains like a lot of parts, right? Why exactly we highlighted those? Because cornea is also important in the technology of eye tracking. It lets the light through, but it also reflects a part of the light. And this is something that we just take advantage of. Then comes the pupil together with the iris right here. So the iris is the colored part of the eye. The pupil is just the opening inside the iris, which lets the light through. The pupil size can change, and this is connected to the light response, but also to cognitive processes, as we all know. But essentially, the pupil is just the opening. What changes the size of the pupil is the iris, which is a set of muscles. Then we have the lens that just projects the image onto the retina, turns it upside down, and projects and focuses it onto the retina. And on the retina, the light converts into the electrical signal and then goes through the optic nerve to the brain. On the retina over here, we have cones and rods, so two types of photosensitive cells. The difference is that cones are responsible for colors and details. Rods are responsible for basic contrast, night vision, let's say, and movements. They're very, very sensitive to respond to a single photon, but they do not provide any information about colors, and they do not give us that many details. The thing is that cone cells are concentrated primarily around this little spot that is called fovea. It's a very little spot with the highest concentration of cones. Peripheral vision is mainly covered by rod cells. Just to show you the graph, right? So this is the distribution of cones and rods around the zero coordinate, if we define it around the center of the visual field. In the center, we have corneas, and this is our foveal area. Sorry, cones we have, like this spike. Rods cover mainly our peripheral vision. If we look at the graph on the right, which shows the acuity of our vision, so basically the ability to sense colors and details, and this graph looks pretty similar to the red graph on the right. It means for us that if we look at something like this store shelf, or it could be your task that you give to your students, we kind of believe that we see all of it, but we don't. So what comes through our eyes looks more like this. We have foveal area, which is relatively small, highest concentration of cone cells. We have parafoveal area, where the concentration of cone cells starts dropping, but basically progressively decreases. And then we have peripheral vision, which is pretty blurry, monochromic, basically shades of gray, so we don't see that many details outside of the parafoveal vision. So that's how we see the world. At least that's what our eyes see. The rest is just processing in the brain. However, this little spot, foveal area, occupies over 50% of the visual cortex of the brain, right? So mainly when we look at something, we primarily process the very central part. Our vision spans up to 220 degrees horizontally and up to 135 degrees vertically. The foveal area size is around 1 or 2 degrees, so it's basically the thumbnail. If you do like this, if you give yourself thumbs up, then this would be your foveal area. And this would be your parafoveal area, the size of the fist, the size of the hand. So how to calculate degrees into physical sizes? If you have a distance of 64 centimeters, let's say, which is usually the distance to your screen when you work in front of your computer, then the size of the fovea will be around 2.5 centimeters. So if you apply the following formula, you can calculate physical sizes. So 2.5 centimeters, that's how much you actively process when you perform your task, when students perform your tasks. So they don't see that much of the rest. Do you have any questions so far?
No, not yet. But you raised some interesting point that what we look at is what most likely what you're thinking about. Yeah, that's really good for research and for mm, pedagogical processes.
Okay, good. So then we need to talk about eye movements and attention, because considering that the foveal area is very small, we need to move the eyes, right, to kind of collect pieces together and reconstruct the view of reality. So that's how it essentially works. So let's talk about eye movements. When we look at a static image, we believe, and we are normally aware of it as a whole. However, what happens when we are looking at it, we fixate somewhere, and then we jump to the next part, to the next part, to the next part, just to collect these pieces together to reconstruct our picture of this image. And in static context, we have two types of eye movements that are called fixations, when we fixate at something, and saccades, when we just jump between fixations. And that's how we interact with static scenes. So what do we need to know about fixations? Fixation length indicates information processing, so the depth of information processing. Typical length is between 100 to 600 milliseconds. There are fixations that are longer, there are fixations that are shorter, but typically will be between 100 and 600. When we are reading something, it will be around 200-300. But normally we make around 3 fixations per second in our everyday life. And this is the time we acquire information, right? We basically process information. And I will go back to the first point again. So if you measure duration of fixations, it could be a valid indicator of the depth of information processing. It's like one of the key metrics. When something is difficult, when you are reading, for example, complicated words, new scientific terms will result into longer fixations compared to more frequent words and that are easy to understand. Those will have shorter fixations. Saccades. So the main purpose of saccades is just go from one fixation to the other. Our vision is largely suppressed during this eye movement. So what does it mean? It means that during saccades, we do not acquire information. So we are almost blind, right? The reason for it is that the main reason is that saccades are very fast. They're fast and ballistic, and they can reach up to 900 degrees per second in velocity. their duration starts with 20 and 40 milliseconds, and then they can be longer. So again, saccades are very fast. We experience some kind of motion blur that gets suppressed by the brain. That is why it's just not efficient to try to get information when we just move from one fixation to the other. So there is some time to plan saccades, and this time is called saccadic latency. And this is at least 150 to 200 milliseconds. And this is longer than the duration of saccades. The endpoint of saccades cannot be changed. So when the saccade is planned, we will finish it, and then we can start a new one if we change our mind. Regressive saccades and saccade pattern can reveal confusions and problems in understanding, right? So regressions indicate confusions. It's pretty clear. When we are confused, we usually go back to check stuff one more time. Cool. So what you see here is a sequence of fixations and saccades. This is a good reading pattern. So that's how we read a text if we know the language. So if I look at this, like obviously the size of the dot indicates the duration of fixations. Those lines between fixations are saccades. Let's do one exercise. If you look at this reading pattern, and the text is in Swedish, so if I look at this picture, if I look at this gaze pattern, I can clearly see that the person is fluent in Swedish. What makes me think so? Why do I believe that the person is fluent in Swedish?
I would guess because he fixated with every word. He tries to look, the focal is to the every word, not jumping everywhere.
Well, some words are not being fixated, like those small words, there are no fixations on them. But okay, anything else that is important here? So there are no regressions in the text. This is like ideal reader. It's not a real recording, in fact. Because normally when we read, we have regressions. So we sometimes need to read the same word one more time, or sometimes read the sentence one more time, if it was difficult for us. And obviously, if the person was not fluent in Swedish, but was, let's say, learning Swedish or learning how to read as a kid, then the person would have more basically a lot of regressions. And this gaze plot does not have any regressions. So the text was easy, and the person is very, very knowledgeable in the language. And this is an important concept for you. So considering that you are doing educational research, you can actually use it as like one of the metrics. Regressive saccades would indicate confusions. Not always, though, because sometimes if you have a task, let's say with a diagram, let's say like you have some text in your task, in your problem, and then you have some diagrams, some pictures, the text might refer the reader to the picture, to the diagram. Then obviously the person would explore the diagram and then go back to the text. This would be a regression. But it's not caused by confusion. It's just because the text referred the reader to the picture. So you need to be a bit careful with it. But this, nevertheless, could be a good indicator of, difficulty of understanding the task if you have a lot of regressions, right? Dynamic eye movements. In dynamic context, we additional to fixations and saccades have vergence eye movements, smooth pursuit, and vestibulocular reflex. Very briefly about those three because it's not your main focus. Vergence eye movements just help us to accommodate for the distance change, so you move your eyes like this to align the foveal areas of the left and the right eye. It prevents double vision. They're slower than saccadic eye movements, and the eyes need to move in the opposite direction. So depth perception, distance perception. Yes. Smooth pursuit is when our attention is on a moving object. So when we want to keep a moving object in focus. Eye velocity is typically less than 30 degrees per second. If the object is faster, when the object is moving faster, we catch up. We move our attention slightly ahead along the trajectory and wait until the object comes into the foveal area. We can pursue only one object at a time. It cannot be triggered voluntarily in the absence of a moving object. So when there is nothing moving in the field of view, we will only have fixations and saccades. We cannot move our eyes smoothly when there is no moving target. So the key takeaway. Let's watch a short video. It's basically how it works. There is a moving target. The person is looking at the target. And you can also see some catch-up saccades. Vestibulocular reflex helps us to compensate for our own motion. So if you move your head or walk, run, jump, the eyes will move in the opposite direction to keep you maintaining your, point of attention. So to summarize, it's more efficient for our brain to focus the attention on the fovea. Depending on whether the object is in motion or static, we will be talking about either fixations or smooth pursuit. And vestibulocular reflex just helps us to keep a stable image. In the periphery, our peripheral vision is mainly covered by rod cells that are very sensitive to respond to a single photon. And this helps us with picking up movements. When something is moving in the periphery, we will most likely, this will most likely get our attention. It's pretty natural. If something is just moving around, for example, out of the window I have horses. Sometimes when they walk by, they get my attention, they distract me, right? I can clearly, even though they are in the periphery here, they get my attention, even though they're pretty far away. It's natural for us, it's reflexive, it's a reflex. And it helps us basically to survive, right, in wildlife. When something is moving, it's potentially dangerous. Which brings us here to covert and overt attention. There are two types of attention. The attention of the eyes, which is called overt attention, and the attention of the mind, which is called covert attention. It's what's going on in your brain. In most situations, it looks like this. So overt attention and covert attention just coincide. But there are situations when you are looking at something and think about something completely different, right? You give your students a task and then just next to them start playing computer games or something else, I don't know, or loud music. Obviously, they will be distracted. They cannot focus on the task. The problem is that with eye trackers, you will be measuring their overt attention. You will be measuring what they are looking at, but they will not be thinking about your task. So you can come to wrong conclusions. So make sure that you have a very, let's say, good working conditions, like minimize distracting factors and make sure that the task is clear. This you will get like a better quality of data, right? Which brings us to the cocktail party effect, which basically describes how it works with our hearing, with auditory cortex, but it works in a similar way for visual cortex. In noisy places, the auditory cortex boosts some sounds to help us to prioritize. So in a noisy place, when we want to hear to a specific conversation, if it's a cocktail party, it will appear louder for our perception. So we will kind of be able to filter it out. People can train to pay attention to things that are important, which is pretty understandable depending on our background, our skills. We will pay attention to different things. People who think that they're multitasking are completely unaware of what they missed. because multitasking is the ability to switch between the tasks rather than to perform them at the same time. Because our brains are wired to focus on one thing at a time. Multitasking declines with age, sleep deprivation, and alcohol consumption. Let's have a look at this picture. A lot of details, a lot of food, some meat, some, I don't know, other stuff, vegetables, sauces. And we will be looking at, if I just ask you to explore this picture, you will pay attention to absolutely different things. It might depend on, maybe if you are vegetarian, you will try to maybe avoid looking at meat or something like this. If you are hungry, then you will be interested in different food. If you are not hungry, then maybe you will pay attention rather to people sitting at the table rather than food, or cutlery or the table itself, because it's a nice wooden table, right? But we will look at this picture differently, and then we will remember different things about it, because if I switch this picture off and start asking you questions, then you will notice that probably you don't remember everything anymore. Do you remember how many white plates were there?
No, I don't.
How many people were there in general?
8 was it?
How many?
8. 4 people on the top and 4 people in the bottom.
Okay. Let's try something else. Was there any fish on the table?
Yes, in the top left, second from the top left.
Okay. Good, good. But there were 6 people, in fact. I will, yes, I will show you the picture. You see, sometimes if it's not important for us, we just filter it out. We just don't care. And that's pretty much like, how our attention works. It's very selective. I will show you this picture one more time and give you 3 seconds to count white plates, right? So 3 seconds, and then I will ask you how many white plates you counted. How many?
5 that are
Five.
bit white.
Okay. Yeah, I agree. If you count this like grayish one, if you include it, then it's indeed 5. Did you notice anything else that was different compared to the first time you saw the picture?
No.
I can give you one hint. It's a photo. So did you notice anything that could not be on a non-edited photo? How about now? So this was the picture you saw the second time. Do you see anything now?
Yeah, it has some added stars.
Exactly. Anything else? I can even give you some contrast, like increase the contrast so that you could see the stars. 5 stars. One of them is in the middle of the plate. So you definitely saw it.
I don't.
So, anything else?
Hmm, anything else?
Okay, so at the bottom I also added some text saying that this is the best training ever in the bottom right corner, and in the bottom left corner saying attentional blindness. Because that's exactly the phenomenon that we experience, right? Our attention is very selective, and it's just to illustrate it, that we cannot, if there are especially a lot of details in the background, then you cannot, like different objects will get our attention, and then we will remember different things about the same background. For example, if I look at like at your room, there are so many things over there, and I will probably not remember everything. I can see that you have some coat of arms or whatever it is that basically gets my attention. I cannot see exactly what it is, but probably some local sign and stuff. And I see that, you have a nice background picture on the left side from me. But You will definitely remember more from my background because I have only one painting behind me, right? There is nothing else that you can see. So you will all remember it. I will remember much less about your background. And that's pretty much like, how different objects fight for our attention in our everyday life. There are also some, so this was as an illustration of how bottom-up processes work, but we also have some top-down factors of attention. And this could be, for example, the task that you give to your students, to your participants. This was illustrated already in the 60s of the last century by Dr. Yarbus in Moscow, who conducted a series of experiments. Basically, what he did was he gave the same picture, the same famous painting of Ilya Repin. He took the same person and then just started giving different tasks to the same person with the same stimulus. And what he observed was that depending on the task, the visual behavior is different. Right? When we have a task, when we have a goal, we will move the eyes differently, which makes sense. And this is something that you should also not forget. So if you want to really compare something across people, you need to make sure that the task is understood in the same way. To summarize, when we want to look at something, we move our foveal area to this object. Eye trackers capture these eye movements and give us information about fixations and saccades primarily after doing some processing. Additional eye movements like vestibulocular reflexes help us to stabilize the image and keep our focus on the area of interest. There is a distinction between covert and overt attention, so attention of the eyes and attention of the mind. Eye trackers do not capture peripheral vision. To get this information, you need to collect surveys or verbal protocols, so you need to ask them in whatever form. The task and the research space conditions will influence the research outcome. Good. Any questions about this part? Because we just covered the background of eye tracking studies on a more theoretical level.
No, it was really it's easily comprehensible for me.
Very good.
Nice.
Thank you so much for the conversation. It was going to cost but can we move to the master data, something like how to design the pilot features for some picture, for example, like which one for every one and two, and how to spin and how to retain it, getting some pilot features.
Sorry, I cannot hear you properly.
Can you repeat again? Yeah. Okay. My question is, the first one, thank you for your basic conception of ice tracking. I want to ask you about how to analyze the data and I mean how to collecting the data from this application because sorry to say, I studied what is completely based in Taiwan, so I know how to getting the data, but sometimes I difficult to use the data. For example, like how to design the area of interest and how to design how to collect the first part seeking duration for example this one. So can you show me the practice how to use this application in Tobii?
Yeah. I mean, we'll get to it. We need to first, cover the we need to first cover the presentation, and then we'll get to the application. We'll talk about it obviously later.
Okay.
Mm-hmm. I just need to transition to the next topic about more technical aspects, and so they just give you information about how eye tracking works first of all, and then about the hardware that you have. So just to make sure that uh, that you have, like, good practices of working after this, because
Thank you.
I think are important when you just start working with eye tracking. Okay, but we'll get to the application obviously later. So about eye tracking and how it works. First of all, here you can see different screen-based eye tracker. This one is the one you have, and this is the one we will focus on a bit more. But before we do so, very briefly about the technology. So it's a camera-based system. It takes images of the eyes, and it also relies on so-called corneal reflection. So our eyes, the cornea, the front surface of the eye reflects the light. And basically what we do is we place some light sources inside the eye tracker. We set them to infrared light because we don't want to disturb people. We don't want it to become obtrusive. But geometrically, infrared light behaves in the same way. So we place light sources and cameras inside the eye tracker. The light sources will illuminate the eyes with infrared light and create reflections on the cornea, like this. Then what happens is, when the cameras will take these images, then they will detect the pupil and the corneal reflection, measure this vector from the center of the pupil to the corneal reflection, feed it into the 3D eye model, and then basically, if we know all the geometry, if we know the relationship between the cameras and the illuminators, if we know where the eyes are, we can just do some math to understand the gaze directions of the left and the right eye to find the intersection point, which will be considered the gaze point on the screen. So that's shortly how it works. There are two techniques that I used, bright pupil technique and dark pupil technique. Let's say when it comes to Tobii Pro Spark, it uses a combination. The difference between the techniques is how you place the light source. If you place it next to the camera, then the pupil will appear brighter than the iris. If you place it away, the pupil will appear darker than the iris. That's how much you need to know, to be honest. Another concept is called eye tracker track box. So do not forget that you are like that. This is like the eye tracker is a camera-based system. So eye tracking can be performed only within certain limited space, right? So you cannot go too far, come too close. You cannot go outside of certain imaginary box. Another concept is finding the right eye model. So here you see different eye shapes, different eye colors. But more importantly, for us is the precise location of the fovea. When we are looking at something, we need to place the foveal area onto this object. So the attention of this person is along this line, not along the optical axis. And we need to kind of measure this angle. And that's pretty much what happens during the calibration. We need to find the relationship between the known position of certain targets, so we know where those targets are in relation to the eye tracker, and we just need to find the relationship between these vectors and the positions of the target. And this will help us to optimize the eye model so that it matches the participant's eyes. What happens afterward is we measure these vectors, we keep measuring the vector, but then we feed this vector into the calibrated eye model, and the eye tracker will return the gaze data, so-called, which is the pupil position, the pupil size, then the gaze point for the left and for the right eye, and average. That's pretty much the data that you can subscribe to. Then the software comes into play, takes this gaze data, and does all the processing. And that's how you can get all the visualizations, metrics, sword fixations out from saccades, and all of this. It happens in Tobii Pro Lab. How to ensure good data quality? First of all, make sure that you position your participants correctly. Before calibrating, make sure that, first of all, they're sitting comfortably. And after this, try to do some adjustment of your screen, of your table and chair to bring them to the center of the track box. And then you start. But they need to sit comfortably. If they don't sit comfortably, then obviously during the data collection, they might just go too far or something like this. To avoid that, first give them the comfortable position, then make sure that they're in the center of the track box. For this eye tracker, it's around 70 centimeter of distance, and the eyes need to be roughly in the center of this tool. These bars underneath indicate basically noise levels. They just take some time window and measure how many gaze samples are successful. If you see that a situation like this, it means that the eye tracker cannot see the eyes. This could be because the person is outside of the track box, or maybe looked away, or maybe the eyes are occluded. There is something on the way between the eyes and the eye tracker. This is the same, but only for one eye. And if you see that the purple bars are flickering and the eyes are blinking, it's most likely related to interference with environmental factors. After the calibration, you will get certain feedback. It's either calibration feedback or validation feedback, which shows how measured samples are distributed around the calibration or validation target. You can do either left and right, or you can have average. After this, we measure two parameters, and those are called accuracy and precision. So accuracy is this distance from the center of the target to the average sample. This is the accuracy. And the precision is the radius of this circle, so the spread, the level of noise. If the accuracy is not good, it's most likely related to systematic error. Try to redo screen setup in Tobii Pro Eye Tracker Manager. Or maybe it can happen that the person who has to wear glasses had taken them off. Then you need to recalibrate. If the precision is bad, it's most likely interference with environmental factors. And this includes the following. It could be related to light exposure. Both light is important because it obviously it's connected to the pupil size, but also infrared light. If you have infrared light sources like the sun, it will definitely interfere with this. It might create additional reflections of infrared light and will interfere with the data. Glasses are fine, but they have to have only one optic power. So no bifocals, no trifocals, no progressive lenses. And your glasses need to be free from scratches and smudges. Right? If there is a scratch in the lens, then the light will go through the scratch and then it will be scattered, creating multiple corneal reflections, which is not something you want to have. So make sure that if someone needs to wear glasses, one optic power, no scratches, no smudges. Obstacles. So nothing should be on the way between the eye tracker and the eyes. When talking about obstacles, we sometimes think about something bigger, but we often forget obstacles that we wear pretty much on us. Could be false eyelashes, too much mascara or makeup when it's really too much. Could be maybe piercing around the eyes. Could be, I don't know, contact lenses with colors. So the change contact lenses that change iris color, and anything else, or even long hair. So if it's on the way, it will obviously be problematic for eye tracking. Some eye properties like droopy eyelids could also cause issues. And a bit more to consider. So this slide just shows you some information about participant management and recruitment. So when you establish a participant profile, we suggest, unless it's the subject of your study, to consider excluding subjects that are likely to cause difficulties. So we talked about optic powers. To add to this, eye surgeries, specifically corneal surgeries, because if there is a scar on the cornea, then obviously it will distort the eye model. so it's not spherical anymore. Eye movement or alignment abnormalities like lazy eyes, strabismus, nystagmus, droopy eyelids, and wet eyes are problematic. So this is something that will cause difficulties. Maybe any questions about participants? All right, let's move on and talk about Tobii Pro Spark finally. So this is your eye tracker. This is a picture of it. It contains one camera in the middle and 3 light sources. It relies on a sequence of dark and bright, as I mentioned. So no specific mode it's set to. All the processing happens inside the eye tracker, which puts pretty low demand on your computer, in fact. Tobii Pro Lab, the software, puts higher demand on your computer. And let's talk about sampling frequencies. So Tobii Pro Spark samples at 60 Hz, which means that samples will be taken every 17 milliseconds. Is it good or bad? It depends on the type of study you are doing. If it's an attentional study, if you will only focus on fixations, then it's good. So it's sufficient, let's put it like this. If you want to do saccade kinematics, then obviously you need a bit more. For educational research, I would say it's totally sufficient, because the main focus of your is attention, this or that way. The latency is around 1 frame, around maybe like 15-ish milliseconds, and this might be only important if you want to try gaze contingency. So if you want your stimulus to update based on the visual feedback, it's like, basically, if you want to try something like this, if you want to program this experiment, when depending on where they look, the stimulus will change. Then you need to keep in mind the latency, otherwise it's not that important for you. In reading studies, it's actually done pretty often. Accuracy. The accuracy of Tobii Pro Spark is 0.45 in the best conditions, so obviously cannot expect it from everyone. And the precision is 0.26 for Spark. So just to remind you what means accuracy, what means precision, right? So we have 4 different situations here. This target shows us both bad accuracy and bad precision, because, like the bullets, if those are bullets and those are targets, then obviously they are just all over the place. This situation shows us bad accuracy because they are far away from the center, from the bullseye, but they're very close to each other, so the precision is good. Bad accuracy, but good precision. This target shows us bad precision, but good accuracy, because they are around the center, but not that close to each other. And obviously, this is good accuracy and good precision, what we want to have. A little bit about system setup. What you need to keep in mind is the position. I guess you would place your eye tracker just under the screen, because the eye tracker is set to track objects that fit within 30 visual degrees as seen from the participant eyes. So if you place it too far away from the screen, then there might be areas that would not fit within 30 degrees. Or if you use a screen that is too big. So there is a limitation. So the screen is limited by 27 inches. I would suggest 24 as I would prefer. 27, it will still work if you place it correctly in the center. Because of this, because 30 degrees is into all the directions, right? If the screen is too big, it might not fit, right? Which screens are you going to work with? What size? So do you have 24 or 27-inch screens or laptop screens?
I think this is 27, right?
27.
Pak ini 24 atau 27? 27. This is a 27, but we did not place it directly under the screen. So there's this is like
Boxnya Tobii di mana? Boxnya Tobii, kotaknya Tobii.
so we have some space below the screen and then to the Tobii. Maybe we should raise it a bit to be exactly under the screen.
But why don't you place under the screen, like there are mounting brackets that are included, like those magnetic
Yeah,
ones?
there's a speaker under the screen. So this is an all-in-one, like what's called.
Then honestly, you need to watch out, because if it's too low, like the top corners might be cut off a bit.
Yeah.
Or you need a smaller screen. Or you need to try with a 24-inch screen, because with 24, it will more likely to work. So you just need to understand it's a physiological limitation. There is not that much we can do about it. It's just that, like, when the angle is bigger, when I'm looking like too far aside, then obviously it's difficult to capture the pupil. It will just not fit within the camera view. So it's not possible. So I would say it's to be expected. If the screen is too big or the eye tracker is too low here, then there might be some error that simply will not track properly. So it's just for you to keep in mind. So maybe 24 is better. Or you need to rearrange your speaker somehow. So if you maybe detach the speaker and place your eye tracker right here, right on the bezel, because you have mounting brackets like those ones, those magnetic ones. You can just stick them on the bezel and use them. All right, let's move on. You need to do screen setup into a eye tracker manager. Just like doing some measurements, and I will show you how to do it. So we can probably go a bit faster. The next thing is that this is basically the size of the track box for Tobii Pro Spark. My suggestion is just simply don't mind these numbers, but rather try to see how far you can go in all the directions. Try it yourself, and then you will understand what's the freedom of movement for your participants. Another thing, we have blink recovery, which is implemented for all eye trackers, so we don't need to lose samples after the blink. And to maybe finish with the presentation, I will show you one slide about the metrics. These metrics have to do with user experience studies, but it's just a good example of why you need to consider quantitative analysis over qualitative, because certain metrics could give you a pretty interesting information, pretty interesting insights. Again, this table is just a drop in the ocean. You can have access to much more metrics, but it's just good to illustrate. You remember when I was talking about fixations, I mentioned that the longer the fixations are, basically the deeper is the processing. How to formulate it. Basically, the longer the fixation is, the more effort we require to process this part of the stimulus. So in other words, when you see that there is a long fixation, the effort is higher. So that's what it indicates. And if you measure average fixation duration, you can probably find out which parts were more difficult to process, or which students took more time to process the same thing, the same formula. So target noticeability and target findability could be measured by or quantified by time to first fixation, right? Obviously, the less time we need to find something or to notice something, the more noticeable or findable it is. Pupil diameter could give us information about emotional arousal or mental workload, if you set it correctly, though, because don't forget about the light interference. And so on. There is much more. So I mentioned, for example, number of regressions and stuff. There are quite a lot of interesting metrics that you can try to incorporate. But it should be hypothesis-driven, obviously, because the same metric could be interpreted in a different way depending on this. We talked about regressions, right? Regressions could indicate confusion, but regressions could also indicate just references, because your text might refer to some other spot. Okay, I'm not that familiar with your common study yet, but maybe do you already have some eye tracking metrics in mind? Other than those, we don't have to talk about those that you see on the screen.
Mm, we only have some that's in the screen that we really want to focus on the interest in when students are doing their exams, and then the cognitive processing in the similarly in exam. But this is giving me some several idea for emotional distress, maybe, or maybe happiness from the pupil diameter. Maybe if the question is too difficult or too easy, people we could measure the students. Um, yeah, their emotional arousal, the distress and the happiness.
Emotional arousal is actually you can do it with pupil diameter, that's true, but you need to like still watch out, because if there is some, like change in the light, or the brightness of your screen change, then obviously it would affect the pupil size more than anything else. So it's not that easy to work with it. So maybe you can try to set it up. You need to have a very good, like, um, uniformly distributed light across the room. You need to have a good control over like your screen brightness and everything. But you might also try to consider other modalities like GSR, for example. Or there are other ways. You can even do it with EEG. Well, not even, yeah, with EEG you can obviously do a lot of things. Um, like heart rate as well. So these things could also give information about emotional arousal. If you synchronize it with eye tracking data, you can actually do quite a lot. Or you can rely on pupil diameter. But just my suggestion is do some literature review before you start, just to see like what other researchers' experience was in the similar field. It's not that easy to work with pupil diameter. But okay, we can definitely get back to it. Let me check. Oops, I can actually stop sharing my screen for a moment. Have you sent me like your stimuli? Because I don't see anything
Uh, you
yet.
can check in chat, and there is a Google Drive link.
Where? In the chat? Okay, I see it. Okay, so if you don't mind, I need to get some coffee. If you can give me like 2-3 minutes, or maybe you can make a short break for everyone. Like, what do you think? Do you need a break, like 5 minutes maybe?
Yeah, okay, okay.
5 minutes would be great.
Okay, 5 minutes then, and then we get back to it. In the meantime, I'll try to download your project, like whatever you sent. Okay, see you in a moment. All right. Let's get back to the training. So the first thing I'm going to do is we will start with Tobii Project Manager. Even if you tried it, I still need to walk you through a bit. Yes. So I have a slightly different eye tracker. I have like an older generation, but it's pretty similar in terms of how it's implemented. Yeah, so this is Tobii Project Manager. I have connected my eye tracker, so we can connect it to one of the USB ports. You can either connect it directly to USB-A or use the USB-A to USB-C adapter. It's detected. I installed all the drivers and firmware automatically. If you haven't done it, well, you probably have, I guess. Now I'm selecting the eye tracker and it's available. It shows me that there is a driver and firmware update. Okay, it's not, but there's definitely a new version of Tobii Project Manager available. I have to stay in this one, but the interface has not changed. So what do we have here? I have a slightly different frequency actually. You have 60 or 30. You can change it here. Sorry, you have a choice between 60 and 33. Here in the frequency, if you click on this one, in the drop-down menu, you can select the correct one. What's important here and what you need to do at least once per screen is the display setup. You see, if I click in this field, I have several setups. When you connect your eye tracker for the very first time, when you attach it to the screen, so let's say use the mount in brackets, stick it on the bezel of your screen. You need to, again, then snap your eye tracker on and create a screen setup here. If I create a new one, you need to choose between regular setup or advanced setup. Regular setup is if you have your eye tracker directly connected to the screen, if it's attached. Advanced setup is when it's not attached to the screen. Which one have you tried, by the way?
The regular one.
The regular. You can do the regular one, but you need to make sure that when you place it underneath, then at least, like, you know, your eye tracker and your screen are on the same surface. It's like there is no distance between them. No, like depth distance. So on the same surface, it could be below, but on the same plane, right? The same plane, not the same surface, yeah. So they need to be absolutely parallel, aligned. Advanced setup, you have to do if your eye tracker is, like, if your screen is way behind your eye tracker. Actually, speaking of, you mentioned like that you cannot connect your eye tracker directly to the screen for like various reasons, but if you have a bigger screen, it is possible to put your eye tracker actually on a tripod and then just move your bigger screen away so that it fit within the angle that I mentioned. Then you need to create an advanced setup. It's a bit more to measure because you need to measure certain distances, like physically the size of the screen, the distance, the angles, offset and stuff. But you need to do it once per setup. So not every time you make recordings. Let's do a regular setup because this is easier to do. And I have a regular setup here because my eye tracker is attached to the screen. So if you go next, you select the screen. I have two screens, obviously you need to select the correct one. Then you go next, and here you measure the distance between the bottom edge of your active display area and the top edge of your eye tracker in millimeters. So if it's below, just measure in millimeters this distance, just to tell your eye tracker where it sits on the screen. Then you go next and need to drag these markers. So you see, I can move them around and you need to align them with white notches on your eye tracker. Have you done this?
Yeah. Yeah.
Perfect. Because if you don't do it, then your eye tracker will wrongly estimate where the screen is and the side of the screen. And then obviously we cannot correctly render the gaze points over the screen. And that's it. Then you give it a name. So I'm not going to save it, but you give a descriptive name to your screen. You see I have like multiple screens here, and the setups are named after the screen. So it's also possible to calibrate here. You don't have to do it because Tobii ProLab has its own calibration routine, but maybe it's good to try. If you calibrate here, there is a different positioning tool. Generally speaking, this tool is used for calibration in two situations. Either you want to troubleshoot something. So, for example, you notice that something is wrong in Tobii ProLab. This is the first place to check because this tool was designed for, like, for the hardware setup, just for the hardware check and stuff. So I'm placing myself correctly, and then I can calibrate myself. So I need to look at the dots until they explode. And now I'm calibrated. So you can see some spread in the gaze data. I can use this calibration. I can even play around with the visualization bubble, so you can see where I'm looking. But that's pretty much it. One more thing that I need to mention here is with those setups could be exported in a file and then imported if, for example, you change your computer. So you don't have to redo them every single time. So any questions about this tool? I think it's pretty straightforward. It's a very basic hardware troubleshooting tool. Make sure that every now and then you check the firmware update as well. If you are connected to the internet, it will notify you that please update your firmware. It's definitely an eye tracker that will get releases, so it's a relatively new one. So please keep it up to date. Let's open Tobii ProLab then. Which version do you have? Do you have the same one, 24.21?
Yeah. We have the same.
Perfect. So this is Tobii. I think you have a different edition, though. I have obviously full edition, internal edition. I don't remember which one do you have. Let me quickly check. Very quickly. Here it is. Because depending on the edition, different project types are available. You have a screen-based edition, just a screen-based edition, right? So what does it mean? It means that among the project types, we have only a screen project available, and then there is in-camera project. The rest is locked. So you cannot do advanced screen, you cannot do glasses, you cannot do external presenter. And I think it's actually totally fine for the purposes you mentioned, because advanced screen project is a table-based presentation which is used in psychology quite a lot. So it's really for more advanced presentation features. Your experiment, at least how I understood it, could be easier designed with the screen project type. A little bit about the starting page as well. Here you can create your project, but make sure you keep your projects locally on your computer, so you don't store them on cloud services or like shared drives and stuff. So it has to be stored locally. We do not support OneDrive or anything like this. Here you can open your project. Your license obviously can be activated and deactivated. It's a floating license. You can activate and deactivate it if you want to use it on another computer. This is the place to check updates. So every now and then we have a new release with new features and stuff, so please also check it. You will get a notification if you're connected to the computer. Here are some useful links and a feedback button if you have some ideas on how to improve this software. What I'm going to do now, I'm going to create a project. I have your file. Oops. I will just call it after your university, the project. And I will change the default location. You can change the default location, but keep them locally. Create. So when I create the project, any project, it will open the project overview. The project oops. I think I selected the wrong one. Let me delete it. I selected the wrong project type. I selected advanced screen, which you don't have anyways. Let me start it over. Screen. Create. All right, project overview. So it has these tabs that you can add. So if you click on these buttons on the left panel, you can add different tabs here. The one that is by default on is the recordings tab, which is empty because we haven't done any recordings. The next one is called participant variables. And this is the place to create your demographics. This usually includes, your demographics. It could be different age of students, different skills they have, gender maybe, I don't know. So if you really have different groups that you want to compare, do you have any participant variables in your planned study? Do you have any demographics?
Student, I think.
The sound is not really good in this. If you use this microphone, the other one was better. So yeah, you can just have I don't know, age or something like this as well, or grade of your students. Whatever it is, gender maybe it's easier. It's always easier. Male and female. Whatever else, it could be skills, anything that you want to just encode in your project. When you have created, do you have any variables to suggest or not? Do you have different groups of participants that you want to compare later on with each other? Or it's not that important?
I think we don't have any classification yet.
Okay, we can always delete it, but when you have participant variables, you can just assign them, right? If you then add a new participant, you can just assign the variables to them, like female or male, in our case. Right, I'm going to delete it because you said that you don't need them. But this is the place for your demographics later on if you want to try, if it becomes important. The next tab here is called Snapshot. It was originally developed for glasses recordings, but you can also maybe use it if you decide to do a screen recording here. It will help you if you have a very dynamic scenario to just remap your gaze data onto something static. I'm not sure if we need to actually focus on this one, but you might want to try it only if you want to show them your PDF as a whole. So that's what I received from you. And this is your PDF that I received that consists of different questions. I don't know what it is. And if you scroll down, you have some, probably math, probably somehow physics, I guess, some physics problem. What was the idea here? So is scrolling important for you or not? Or you just wanted to show them, like page by page. When they're done with the first page, then they continue with the second and they don't have to scroll anything. What was the idea here? So how did you want them to interact with this? Do you want them to...
We want to the participants scroll it and do the problem, do the and answer it just in their mind.
So this
Do you
PDF,
get it?
again, like, what's the idea to just give them this PDF and then they are supposed to click on the buttons or something like this? Or what was it supposed to be, like some HTML file or some other executable file for them to test? Or you just wanted to show them picture by picture without scrolling? So this is my question. So there is a question. They probably need to press the button or answer it. In what form? Pen and paper, verbally, clicking buttons, or like what's the idea?
Clicking button and then uh, the screen will be moved to
Go to the
next.
next. Okay, I see. It's here as well, some buttons. Here we have buttons. And what about this one? What do they need to do with that?
Uh, we want to see interval of interest and which point that have longest duration.
But how do
Uh,
are they supposed to solve the problem? Like, on what?
yeah, so we are kind of, we want to measure in how many percent of the time of on area of interest. For example, are they more interested in the story or are they more interested in the numbers, or if they could not solve the problem, where could they, are they going to read it again and again and again, or how? Maybe that's one.
But okay, like, do they need a pen and paper to solve the problem, or are they supposed to give an answer verbally because there is nothing? I mean, here I see some like buttons that they could click potentially. But what about this one? Like, what is just to read or to answer some questions or to solve some equations here? Like, what's the task you give them?
Yeah, this one, the last one, we
Yes.
just confirm the participants supposed to have a pen and paper,
I
and
see.
they're doing. We also measure which how many percent of the time they are focusing on the problem and off the screen to the
Yes,
paper.
I see. I see. Okay. I just wanted to mention that obviously when they are looking down, then the eye tracking data will be lost. We like do not do it, right? So it's expected. For example, you have some fixation here, then there is a saccade going down. It means that they switch to the paper, but you will not know the coordinates of the fixation. So it will just indicate that they are looking outside of the screen. Okay, I have some ideas for today how to design it. Obviously, there are still options, and you can probably later on try a different approach. First of all, I need to break this down to pictures. I need to convert this PDF into pictures. I don't think it's possible in one go, so I'll probably need to crop some snapshots. Let's see if I can do it in an easy way. I don't think so, to be honest. Let's try. Save as. Nope. I can't really do it.
We will send the picture.
Oh yeah, if you have pictures, please do. Yeah, it will be easier. I will keep the PDF as well because I can use it, like, to maybe show you screen recording if we have time. If we don't have time, then we do it next time. So I'm like, just let me know when you've sent your pictures. In the meantime, I will just go on and continue some explanation. So where were we? We were on the snapshot side. There is also a possibility to create events for behavioral coding, and this can help you to log some behaviors. Anything. For example, when they took their pen, when they started writing something, when they said something, any behavior that is important for you. You can just create events here and assign hotkeys to them. Right? And these events you can then log when it happens. When you're moderating your recording, you can just log those events. Like, they started writing, they said something, they did that or that. Certain events will be registered automatically by the computer, like key presses and mouse clicks we log automatically. The next one is called stimulus variables, the next section, the next tab. Stimulus variables you can use if you have different categories of your stimuli. For example, if I look at your PDF, right here.
Sir,
Yes.
we have sent the picture in chat
Perfect.
like before. Yeah.
Perfect. I will just go ahead and download it then. Perfect. How do I download all of them in one go? Let's see. Let's see. Nice. Now I have it. Oops. Cool. That is very good. Okay. PNG. Perfect. So yeah, stimulus variables can help you with some context, let's put it like this. In your case, I can see that you have two types of stimuli. You have questions, or let's call it task. I don't like the word type. You have questions, and you have problem. It's not necessarily to log them. I just need to show you some software features as well, right? That's why I'm coming up with some ideas. Let's go to the design tab and design the study. Do you have any questions about the project overview, by the way, about like all these things? Okay, well, I think it's pretty straightforward. So what do you have here? You have a timeline already that is empty, and you need to populate your timeline with stimuli. That's pretty much how it works. Timelines could be duplicated. When you were sharing your screen, I noted that you actually have multiple timelines here as well, right? Timelines could be then duplicated, or you can create a new timeline. Timelines, you get name accordingly. You can change the name of the timeline. By default, it's just timeline 1, timeline 2, and so on. You can change the name. Another thing that I mentioned right away, the resolution of your screen. Please set it correctly. By default, it's set to Full HD 1920 by 1080. If it's different, the resolution of your screen that has your eye tracker, so your presentation screen, it should match. If you change it, for example, I have let me make it a bit bigger. I have my calibration element. If you change the resolution, the aspect ratio, then you see it will also change accordingly. So this is important. Okay, you can populate your timeline with stimuli, and you can also select different stimuli on the timeline. When you select, let's say, this calibration element, the properties panel will adjust accordingly and show you the properties of this element. What properties the calibration element has? First of all, you can choose to validate your calibration or not. You see validation is off. Those 4 targets in the middle are to validate the calibration. You can change the number of targets from 2, 5 to 9. Honestly, I think 5 is optimal. You don't need to set it to 9. Then you can choose between point calibration video or size and video. Point calibration works for adults. Video and resize and video works better for very young kids, for infants. This is the only difference. Another thing to pay attention to, and it's especially important if you wanted to try pupillometry, but not only. If I open your pictures, or maybe the PDF for now, I can clearly see that there is a white background and maybe some black text on it and sometimes like blue buttons. The background is white. And it will really perform better if you change the background of your calibration target to white, if you also calibrate on the white background. And the target calibration you can set either to blue, because you have some blue buttons, or to gray maybe, or to black. I would actually go with maybe like something like this, bluish. And then it kind of matches what you have on the slides, on the pictures. So please change it to either gray on white or black on white or dark blue on white, something like this. If you have any specific preferences for the color, you can just enter the hex code of your color. If you know exactly what it is, you can change it, like you can check it in every graphics editor. Let's populate the timeline now. You can add a picture, you can add a video if you want. Then you can add groups. Groups will help you with randomizations and presentation orders. If you want to randomize your presentation order or just repeat something, right? So I don't think it's needed in our case. To be honest, in general, it's a good practice. It's a good, probably, like, habit, I would say, to still keep your main sequence in a group. So there are certain advantages to it, because sometimes maybe you want to add something at the end, like an instruction element or something else that is different. It's just a good habit. I don't know. Like, I kind of like having my presentation order in a group, even though you don't have to. But if you at some point decide to randomize your like questions and stuff, you can just apply one action to your group, and then the order will change. Like I said, it's up to you to decide whether you have your pictures in the group or not. But it's maybe a bit easier to handle. If I add a group here, I can give it a name, for example, main task, and I will show you why. Let's go through the elements. The next one is called web element. If you decide to give them a task in HTML format, for example, or anywhere like on the internet, then you do a web recording. It has certain features for web recordings. The next one is called screen recording. If you add a screen recording, it will just record your screen. If scrolling is important, if they really need to physically scroll your task, you can just display your task in this PDF format, and then when the timeline approaches the screen recording, it will just record the screen. Whatever is happening on the screen will be recorded. It's for more advanced application testing, for example, when you cannot use pictures, when you don't want to be limited by just pictures and videos, then you do screen recording. It's more challenging to do the analysis, but you can try if you want to. The next element is called text element. If you add a text element, I can, for example, bring it before my task. It's a very simple graphics editor. You can change the background to white and you can type your text. Like, please answer the questions by pressing the buttons or something like this. Press space to start. So we have this text element, we have the group, and after the group we say thank you. If I select the first text, you can choose how you advance. It could be time, it could be key press, it could be mouse click. I would say that the instruction, let's give it a name. I will rename it. Right here, instruction. I want to advance on a key press because I instructed them to press space to start. Right? That is why when they press space, the task will start. So we advance on a key press. If I select my thank you element, you can call it thank you. I will maybe advance on time because I don't want them to do anything. We can just give them, I don't know, 5 seconds to display thank you, and that's it. It's also for us, for moderators, a good indicator that the task is over, especially if you do randomization. Again, you don't have to work this way. I'm just giving you some ideas for the future, maybe for your future experiments. The group, into the group, I will just add my pictures, or your pictures in this case. You can just drag and drop them, basically all of them. I think the order is wrong, right? This was supposed to be the first, the second, the third, the fourth, the fifth. What was next? This one. Make first this strong man, so one, and then the mathematical problem. Perfect. As you can see, the resolution of this pictures is higher than the resolution of my screen. You see, it doesn't fit. And if I open your pictures here, we can always check, right? If I right-click on them and check properties, where was it? No, not here. Properties of the picture. Details. So the resolution is 2000 by 1414. My suggestion is, for the future, and to make it nice for the graphics card, change the resolution of the pictures, to 1920 by 1080, or if your screen is set to this resolution, then obviously you need to change the resolution of your screen. Well, we don't even have this one. Alternatively, you can, again, it's always better if the resolution matches for different reasons. It just looks more natural, so, and it's also better for the graphics card performance. A quick fix here would be if I click on Control A within the group, right, to select all the elements inside the group. Now I can change properties to all of them. And I will just change the scaling from original to fit to the screen. Like I said, so considering that the resolution of the pictures does not match the resolution of the screen, we have some margins here, right? So it will not be displayed completely full screen. There is some, you see some like black kind of stripes here on the left and on the right. And that's exactly how it's going to be displayed, right? So we have some black margins. The easy fix, again, I'm talking about the easy fix for now, but ideally, the good habit is to match the resolution of your pictures with the resolution of the screen. But the quick fix would be to change the background here to white, right? Background color, I change it to white for everyone. So Control A, background white. Now they look a bit better. Now that's how they are going to be presented. We don't have these like black stripes to the left and to the right. By the way, you can also change positioning. They don't have to be presented in the center. I don't think it's needed. And another thing, if I click on Control A again, how do I want to advance? Maybe if I exclude the last one, I want them to click on the answer. So I will advance not on time, I will advance on mouse click, because it's expected that they will click on the answer, right? Except for maybe this one. Here, we don't have to click. So this one, you can either advance on time if you want to limit them by a certain time, or you can also use mouse click or key press. Up to you. Or you can choose all of them, whatever you prefer. You can even advance on gaze. It's not the case here, but you know that you can advance on gaze. If you advance on gaze, you just need to define some trigger zone over here. Like for example, you expect them to look within this window for 300 milliseconds by default, but you can change it. When they have looked here, then the task is over. Again, I'm just giving you some, I'm just walking you through the software for the future. I think we can keep both, key press and mouse click, because it doesn't really matter for the last one. The rest will advance on strictly mouse click. Does it sound reasonable to you, or do you have any like suggestions?
No, I think that sounds good.
Then I will again select all of them except for the last one and assign stimulus variables. The first 7 ones will be questions. Last one would be problem. That's it. It will just help you to, you know, to find them better in the data later on. Okay. You can see that you, yeah, you send a message in the chat. Do you want to comment on it? So again, like, yeah, I can see that you wrote a message about questions. So what is it about in the link? Hmm? I guess you are suggesting like 3 types of questions, right? And you want to categorize them by concept questions, reasoning questions, and problem solving. Is that correct?
Yes, this is correct.
Let's do it, yeah, maybe it's better. So I'm changing the stimulus variables, and then I will assign them accordingly. So the first 5 are concepts. You said concept, perfect. The second one is reasoning, and second 2, and this is problem solving. Perfect. Problem solving, yes. Reasoning, concept. Okay, very good. Okay, any other like questions or comments here? Because we're pretty much done with the design. We can make a recording. Like, I mean, I will make a recording. And by the way, like we're trying to do the same in the meantime, or not, or just listening.
We just listening.
Okay, I will probably send you this project after the training. You can collect some data with it, or you can try to design it from scratch, or design something similar based on what you heard. I'm going to make a recording. Oh yeah, stop. I wanted to actually add validation. I want my validation back here. Cool. Now I'm done. I think now it should work fine. Over here, first of all, the eye tracker, make sure that the correct eye tracker is selected, the sampling frequency is correct and stuff. If you click on the plus button on the device ribbon, you can add a participant camera, a microphone, or Shimmer GSR. Shimmer GSR is if you want to do galvanic skin response together with your eye tracking data, if you have it. You can also record a sound if you want to communicate with your participant. You can also add a participant camera. If you add a participant camera, this would actually be probably a good thing to do, because if you add a participant camera, you can clearly see that when you are losing the gaze data, that they are looking down or writing like something, right? To justify the loss of data. If you use a participant camera here, if you add it, you can add a participant camera. Well, now it's busy, my camera is busy with the session, but you can also film them. And now my eye tracker is disconnected. Very good. Let me reconnect. Cool. Another thing, I have 2 screens. I will actually share both screens with you. You can see two screens now. I need to select the presentation screen. So in our case, it's the screen with the eye tracker. How do I know which one is where? I click on the identify button. You see that on the left, I have the first screen, on the right, I have the second screen. I kind of want to present my stimuli on the first screen. That's why I need to change it and drag my presentation away to the second screen, because that's how I want to arrange them. I will prepare my timeline because I have some text elements. I will create a new participant. You can assign variables if you have them, and after this you click on Start Recording. So I need to position myself at around 70 centimeters. Now I'm ready to calibrate. And now to validate. Cool. So here we have our calibration and validation feedback. You can check the accuracy and the precision. If you hover the mouse cursor over individual targets, you can have it individually or on average. For my tracker, it's pretty good. I will continue. Actually, for Spark, you will also have similar results. Now I will continue. Please answer the questions by pressing the buttons. Press space to start. I'm pressing space. Oops. Where's my mouse cursor? Sorry, I think I need to stop the recording. Let's check the design. Yeah, I forgot to show mouse cursor. Control A, show mouse cursor. The good thing is that if I now select the same participant and start the recording, I can reuse calibration because I just calibrated myself. So it will just pull the calibration of this person. Now I can continue. I don't know what to answer, I just want to do some clicks. So you see when I'm clicking, so it just brings me to the next picture. Whatever. Oh. Well, and same here. I can just press space, for example, alternatively. It probably requires more time, but yeah. And now after 5 seconds, the task is over. And then I'm going to save this recording. Do you have any questions about it? Because I think that's pretty much how you make recordings. So when you tried the eye tracker, so did it work for you with recording? Did you run into any issues or how was it?
We have run the eye tracker and we get some data, but
Mm-hmm.
uh, we will try another stimuli.
Okay. So what I can do again, I can send you this project if you want to make some recordings with it, or maybe you want to modify it in whatever way. Maybe you want to design something similar, try it yourself. Maybe you can also try with a screen recording if you prefer, with the PDF, or maybe like some other application. Then I will ask you when you have made some recordings, you send them back to me, and we use it for the second session. So you can either design your own project, modify the one that I send you, maybe create a new one, and then we use it.
Yeah, I think that sounds great.
For today, that was pretty much it. What I wanted to show you, we need to decide when we have the next session. Do you already have some ideas? Do you have some preferred time or so, or dates, or time frames?
Yeah, I think we could not decide today because we do not know the, the availability, but we will contact you regarding that.
Well, don't wait too long because like we only give usually 6 months to deliver trainings. I can extend it a bit, but yeah, we cannot wait too long. And again, it doesn't have to be like your study. You just make some pilot recordings. That's it. I don't need anything like that because I still need to walk you through the analysis, right? We haven't touched the analysis. So the session will be more focused on the analysis of your data or how to do the analysis rather.
Yeah, I think it should be months.
Okay, perfect. Yeah, just let me know, just contact me and we will find a date that works for everyone. Probably the same time. Also 2 hours. Good then. Then, if you don't have any questions, I would thank you for your attention. And well, I will send you a follow-up email later today, I think.
Thank you very much, Mr. Kiro. It was very enlightening to say the least.
All right. Thank you. So let's stay in touch then. And yeah, good luck with your recordings, with your data collection and stuff. Goodbye.